Start this interactive lesson right by downloading your Microbiology It workbook. Click the link in the description box and let's microbiology it. The oral cavity hosts one of the most diverse microbial communities in the human body, encompassing bacteria, fungi, and archaea. Environmental conditions vary between surfaces. Saliva provides a low nutrient, fluid medium containing antibacterial enzymes such as lysozyme and lactoperoxidase, while the teeth and gums offer nutrient rich solid surface ideal for colonization. Microorganisms form structured, cooperative communities known as biofilms or dental plaque, which enable them to resist mechanical removal and antimicrobial agents. Within these biofilms, metabolic cooperation among species facilitates nutrient exchange and environmental stability. Streptococcus represents the most abundant genus in the oral cavity, followed by Veillonella, Parvula, Actinomyces, and Fusobacterium. The oral microbiota is dynamic, influenced by diet, hygiene, saliva flow, and immune function. Although most species are commensal, ecological imbalances can promote opportunistic infections, including dental caries and periodontal disease. The oral cavity thus acts as both a microbial habitat and a defensive barrier, reflecting a delicate balance between microbial coexistence and host protection. Are you ready to micro mind map? Well, let's get started. We are going to micro mind map oral and airways microbiota. Our first branch is oral microbial ecosystems. We have environment, biofilm, and dominant genre. Now let's go back up to environment. We have the teeth, gums, and from there we have it's nutrient rich. And then stemming from environments, we have saliva, which there is poor nutrients, and enzymes. On to biofilm. It's structured and it's a community. And it protects microbes. Onto dominant genre, we have Streptococcus, Veillonella, Parvula, additional microbes, Actinomyces and Fusobacterium, and Haemophilus. Microbial colonization of the mouth begins within the first days of life. Before tooth eruption, the infant's oral microbiome is dominated by aerotolerant anaerobes such as Streptococcus and Lactobacillus. The eruption of teeth marks a significant ecological shift. The creation of non shedding surfaces allows the establishment of complex biofilms containing strict anaerobes. Distinct oxygen gradients form within the mouth supragingival plaque, which is above the gum line, remains oxygen rich, and supports facultative species like Carnibacteria matrushati, while subgingival plaque, which is below the gum line, is oxygen depleted, favoring obligate anaerobes like Treponema sokransky. When pathogenic anaerobes proliferate, inflammation, gingivitis, and tissue destruction ensue. Our second branch that we're going to move on to is oral microbial development. In the early life, pre-teeth, aerotolerant anaerobes are present like streptococci and lactobacilli. So, what happens after tooth eruption? Well, there's an increase in anaerobes due to the formation of biofilms and oxygen gradients. Our next branch is subgingival plaque. There's low oxygen environment here, which supports, of course, anaerobes. Then we have supraginval plaque, where this environment has more oxygen and supports facultative species. The respiratory tract is divided into two major zones: the upper respiratory tract, which includes the nasal passages, the pharynx, and the larynx, and the lower tract, which includes the trachea, bronchi, and the lungs. Now, the upper tract harbors a diverse but controlled microbial population, including staphylococci, streptococci, and diphtheroid bacilli, and gram-negative cocci. Mucus secretions trap inhaled microorganisms, which are either expelled by sneezing or coughing or swallowed and destroyed by the stomach acid. Some potentially pathogenic bacteria, such as Staphylococcus aureus and Streptococcus pneumoniae, can persist harmlessly in the nasopharynx of healthy individuals, creating a carrier state that becomes clinically relevant when host defenses are compromised. In contrast, the lower respiratory tract is typically sterile due to robust defense mechanisms. Ciliated epithelial cells line the trachea and bronchi, propelling mucus and trapped particles upward in a process known as the mucociliary escalator. Only particles smaller than 10 micrometers can bypass these defenses to reach the alveoli, where infection may occur. Onto our last branch of the oral and airways microbiota, we have airway microbiota. Two branches stemming from it. We have the upper respiratory and the lower respiratory. Now let's tackle upper respiratory. So we have mucus and ciliary action. That's where pathogens and microorganisms and particles are trapped and expelled. Common inhabitants are staphylococci, streptococci, and diphtheroid bacilli. Potential pathogens are Staphylococcus aureus and Streptococcus pneumoniae. They can colonize the nasopharynx and cause no disease for the healthy carrier. On to lower respiratory tract. Included in it is the trachea, bronchi, and the lungs. It is protected by ciliated epithelial cells, so that's our second branch, and it is sterile. So the lower respiratory tract is sterile. Across the gastrointestinal, oral, and respiratory systems, microbial symbiosis revealed shared ecological and physiological themes. In all regions, microbial colonization depends on local nutrient availability, oxygen tension, and host defenses. Anoxic or low oxygen niches, such as the colon and subgingival plaque, select for obligate anaerobes, while more oxygenated surfaces like the oral mucosa favor facultative species. Biofilm formation is another unifying strategy that enhances microbial persistence, nutrient sharing, and resistance to environmental stress. These stable microbial communities play vital roles in nutrient cycling, immune modulation, and protecting against invasive pathogens. Host defense mechanisms, including mucus secretion and antimicrobial enzymes, epitheliocele, and immune signaling, function to contain microbial populations and maintain equilibrium. When this balance is disrupted, opportunistic infections or systematic inflammation may result. We have our micromind map for host microbe dynamics. So our first branch is shared functional themes. Stemming from that, we have anoxic niche, biofilm formation, and host defenses. So let's go back to anoxic niche. We have obligate anaerobes are promoted for growth. Stemming from biofilm formation, we have stability and protection, and the host defenses, the mucus, cilia, lysozymes, and immune tolerance. Disruptions to the microbial equilibrium or dysbiosis have significant clinical implications across body systems. In the gastrointestinal tract, dysbiosis is linked to inflammatory bowel disease, obesity, and metabolic syndrome, often due to a reduction in beneficial anaerobes and overgrowth of pro inflammatory species. Similarly, in the oral cavity, shifts in microbial balance contribute to dental caries, gingivitis, and periodontitis, conditions that can seed systematic inflammation and cardiovascular risk. In the respiratory tract, colonization by opportunistic pathogens such as Streptococcus pneumoniae or Haemophilus influenzae can progress to pneumonia under immune stress. Understanding these microbial ecosystems provides a foundation for targeted therapies such as probiotics, prebiotics, and microbiome restoration. Our second branch of the host microbe dynamics is clinical connections. Now, this is all about disruption of balance. What disruption of balance is disease. So we have the GI dysbiosis, and stemming from that, we have colitis and metabolic disorders. Next, we have oral imbalance, and stemming from that, we have tooth decay, gingivitis, and periodontal disease. And the last branch, stemming from clinical connections, is respiratory imbalance. When there is an overgrowth of carrier bacteria, the host can get pneumonia. Do you have a healthy understanding of oral and airways microbiota? Now it's time to test your knowledge with a few problems. If you haven't already, download your workbook, which has notes, 20 microbiology problems, and the answers, then click the link in the description box to download it now. Now it's time. Feel free to pause the video and place your answers in the comment section for these problems. If you found this video helpful, then like, comment, and click on the next video.